Hello everyone, in this video we will discuss about packed cell volume or hematocrit. Now what is packed cell volume? So whenever we get a EDTA sample of the blood, if we centrifuge it, the volume of the red blood cells, this is the packed cell volume, this is the hematocrit. Okay, and we will discuss this in detail. So what is packed cell volume? It is the volume occupied by the red blood cells when the sample of anticoagulated blood is centrifuged. Now what does it tell us? It tells us the relative proportion of the red blood cells to the plasma, and it is also known as hematocrit. And the units of expressing this is percentage. It is percentage of the original volume of the blood, or it can be a fraction. Okay, now going to what is the use of PCV. So we already understood that whenever we centrifuge the blood is there is red blood cells, then there is plasma, and in between there are WBCs. So what is the use of getting this volume of RBCs? Firstly, this helps us in detection of presence or absence of anemia or polycythemia. Now, if the anemia is due to decrease in the RBC cell count, then the PCV will be low, or either there is polycythemia in which there is increase in the RBC cell count, then also PCV will be useful. Secondly, it is also useful in estimation of red blood cell indices. I have a separate video on red blood cell indices. So for calculation of MCV and for calculation of MCHC, we need the PCV, so it is useful in that also. Secondly, also we can recheck the accuracy of hemoglobin value. We do hemoglobin by other methods like sign hemoglobin method, but to recheck the accuracy we can also check by this method. If we multiply hemoglobin into 3, we get the PCV. So we can if we have PCV, we can take an idea that how much the hemoglobin of the patient should be. So this was a use. Now there are two methods how we can estimate hematocrit. There is a macro method, and there is a micro method. Now macro method, it requires more amount of blood, so it is known as macro method. The main method, it uses the Wintrobe's tube, which is also used for estimation of the ESR. So if we have a patient in which who ESR and PCV both we need, we can use the Wintrobe's method. And secondly is the micro method, in which the this is known as micro hematocrit method, because in this a very minute quantity, very less amount of blood is required. So mostly the micro method is preferred because it is rapid, it is convenient, and a very less amount of blood is used to determine this. Okay, now we are not discussing in detail about the method. First, you can see over here, this is the PCV. If you see, this is the red blood cell, this is the PCV, and then we have a separation into plasma, and then there is a buffy coat. Now buffy coat contains mostly the WBCs, the platelets. Okay, now whenever we centrifuge the anticoagulated whole blood, there are three zones which we see in the Wintrobe's tube. There is a plasma, then there is buffy coat layer, then there is packed cell, packed red cell, and then we can measure the packed red cell, and this is the PCV. Now buffy coat is especially important if we want to check something which is in rare quantities, such as uh, LE cells, malarial parasite. Especially if the malarial parasite is very rare in the blood, we can estimate, we can check from the buffy coat layer also. And if they are immature cells, they can be present in the buffy coat layer. So for this it is important. Now importance, whenever we centrifuge it, we get plasma. So plasma normally it is straw colored. However, it is colorless in case of iron deficiency anemia. It's pink if there is any hemolysis is present, because the RBCs will break down, it will give a pink hue. Yellow if there is serum bilirubin is raised, and in hypertriglyceridemia the plasma can be milky. This is not in relation, this topic is not in directly relation to the PCV, but because we will centrifuge it, then we should also check about the plasma in addition to the PCV. Okay, now significance of PCV. What is it? Firstly, we already discussed that in anemia, the PCV will be lower. Mostly, it will be lower, and PCV can be increased in. It can be increased in whomever the plasma is decreased, like dehydration, shock, burns, and lastly, if the RBC count is increased, like in polycythemia. So, the significance of the PCV comes mostly in the anemias, in the polycythemia, and the disorder of red blood cells. Now, going to the micro method. Now, micro method, we are not discussing in detail the procedure, but what we do is we take anticoagulated whole blood, mostly the capillary blood only. We take it in a capillary tube. It's a very narrow tube, and we take it in that, and now centrifugation is done. Now in centrifugation it is done in a special hematocrit centrifuged, and then we take the reading using the microhematocrit reader. Now this method is also very accurate and is mostly used. Okay, so you can see over here, these are the microhematocrit hematocrit tubes, capillary tubes. These are used, and you can see how much thin they are, so they require very less amount of blood. So we can use these also. So going to the reference ranges, it varies according to the physiological condition of the person. It varies according to the males and females. Firstly, like in males, 40 to 50% is the PCV, and in adult females it ranges from 38 to 45%. But if you change the physiological condition, if the female is pregnant, now the PCV will be lower. It will be 36 to 42%, and this is very much of importance, because in adult females, when they get pregnant, the plasma it increases more than the increase in the RBC count. So the PCV is slightly decreased. Now in children, it's around 37 to 46%. We are not discussing in detail, but newborns, they have a higher percentage of PCV. They have 44 to 60%. So this was uh, about the PCV. Going to the journal notes, journal important things which can change the PCV. So firstly, if during blood collection, if there is prolonged application of tourniquet, it will increase the hematocrit. Also, if there is excess squeezing of the finger during the skin puncture, this will dilute the sample with the plasma, with the tissue fluid, and this will lower the hematocrit. Also, whenever we have to determine the PCV, we have to ensure that blood to anticoagulant ratio should be proper. If we have excess of EDTA, this will cause shrinkage of the red blood cells, and it will again falsely lower the hematocrit. So these are the things which we have to take in things we have to think about it if there is PCV is deranged. So low hematocrit can also be there if there are clots in the sample, and if the RBC morphology is changed, like in microcytosis, macrocytosis, spherocytosis, and sickle cell anemia, so what happens is plasma gets entrapped within the RBCs, and there is artifactual rise in hematocrit. So, if the RBC morphology is correct, we can report the hematocrit as it is. But if it is varied, we have to check whether any sickle cells are present and spherocytes are present. Okay, so this was all about the PCV or hematocrit. So if you like this video, do like, share, and subscribe to this channel, and ask any queries regarding this topic. Thank you.